So we talked about proteins, carbohydrates, nucleic acids. All of these are organic molecules or biological molecules. But I'm going to end this series of organic molecules slash biological molecules with lipids because they're also organic slash biological molecules. And why do I mean by this? Simply that these molecules, without these molecules, life would not be possible on Earth. And they're major components of living organisms, including yourself. That's why you need to learn them for classes in medical school or other classes if you're studying biology in general. That's why you learn about lipids. And I have some roles here, a list of roles that lipids play in your cells that can actually give you an idea of what I'm talking about, of their importance in your cells. And the first role is that they are energy storage molecules. And if you look at lipids, they're able to form and break bonds. And when they do so, they're able to either release or store energy. And you're going to learn about that in more detail in classes such as biochemistry. But for now, have an idea that lipids are clearly energy storage molecules, okay? The second thing to know is that they're major components of cell membrane. And if I draw a cell here, I'm not a good artist, but anyway, what I want to have here is a simple drawing of a cell with a nucleus. And this structure here surrounding my cytoplasm is called a cell membrane. And the cell membrane is comprised of many things, including a phospholipid bilayer. And a phospholipid bilayer or phospholipids are clearly in this group, in this class of molecules, the lipids. And we're going to learn in a little bit more detail. We're going to talk about this structure, the cell membrane, and you're going to see what I'm talking about. But for now, understand that this really, really important structure that surrounds the cytoplasm, that encloses the cytoplasm, is comprised of mainly lipids, okay? The third thing that I want to talk about, the third role that lipids have, is in cell signaling. And what is cell signaling? Think about it. Cells need to communicate with one another. Because if there is a change in the outer environment, for example, cells need to adapt, let's say, in a way they need to cope with that change in the environment. And they do so by communicating with one another. They send these molecules that, let's say, warn them of the change of the environment, and that way they're able to change their inner environment and that way cope with what has changed in the outer environment. Okay, this is a very simple way to think about cell signaling, and we're going to talk about it later in other tutorials. But for now, know that lipids are part of the structures of the molecules that are used as messenger molecules between these cells. They use these molecules to communicate with one another. And there are some called steroid hormones, and steroids are definitely lipids, and they have a very interesting structure that we are going to discuss also later on in this tutorial called sterine. It's a very cool structure. You're going to learn a little bit about it. We're going to learn a little bit about it. But for now, just keep in mind that there are steroid hormones which are used in cell signaling, and steroids are definitely lipids. Two last things I want to talk about lipids in this slide is that they're highly hydrophobic. And you probably know the definition of hydrophobic. Hydrophobic means that doesn't like water. And to say that lipids do not interact with water molecules well. And if you look, this is basic chemistry, but if you look, water is a great solvent. And why is it a great solvent? Because it has a positive side and a negative side. So it's quite polar and it's able to interact with other polar molecules. But lipids have a very, I was going to say weird, but not weird structure. They're comprised of long chains of carbons, which make them kind of non-polar. And because they're nonpolar, they can't interact. They don't have the negative or the positive side where water molecule or water molecules can go in and interact and be, the solvent for this type of molecules. That's why they are only soluble in organic solvents. And organic solvents are definitely nonpolar. So, and that's why they interact well with lipids, because lipids are also nonpolar due to the long chains of carbons. So I talked about lipids, and I gave you a basic description of what these molecules are. But for biology or molecular and cellular biology, I want to give you just the basic description or classification of these molecules, and you can find different ones out there for lipids. And the first one I want to talk about is triglycerides. And as the name indicates, tri, meaning we're going to see three things. And the three things we're going to see are these three fatty acids that you find here. So these are three fatty acids, and they're going to be bound to this structure here, which is called a glycerol molecule. So a triglyceride is glycerol. So a triglyceride is three fatty acids bound to a glycerol molecule, as you can see here. And these three fatty acids, and look at your organic chemistry notes because you're going to see some more detail there. But for biology purposes, all I want to say about these fatty acids is that they can vary. That's why I put different names here, different letters and numbers, R1, R2, and R3, because they can definitely vary according to the oil, according to the bonds between these long carbon chains here. They can have saturated or unsaturated molecules, and you can see that in more detail in your organic chemistry note. But for now, just keep in mind that triglycerides are these glycerol molecules that bound to three fatty acids. And these are seen in your cells accumulated as fat, liquid droplets in the cytoplasm of your cells. And this is how energy is actually stored. It's time to talk about phospholipids, and I have a representation of these molecules right here on the left, and it looks complicated, but it's not. What you are seeing here is a choline. So this is a choline, and then you find a phosphate group right here. And the phosphate group is attached to the third carbon on the glycerol molecule. So this is a glycerol molecule. And you saw a glycerol molecule attached to three fatty acids in triglycerides. But on phospholipids, you're going to find only two fatty acids. And on the third carbon of the glycerol is here. This is the carbon that is bound to the phosphate group. And because this happens, you see a phosphate group attached or bound to a glycerol molecule. These can also be called glycerophospholipids. Makes sense, right? You see a glycerol molecule bound to a phosphate group. And what I want to also explain here in the phospholipids is that from basic biology, where you learn that the basic structure, the simple way to represent a phospholipid is a one head attached or bound to two tails, which is what we see here. Say this is the head right here. Okay. I would probably include the glycerol, but anyway, and then you have the two tails. And what I can show you on this head and two tails is that you find in or on the head, you find a positive charge right here and a negative charge. So one positive and one negative side of the head of my phospholipid. And that means that this side is it's hydrophilic, meaning that because you find two charges here, it's going to interact very well with water because water also has two charges, one positive and one negative on the oxygen. This is basic chemistry. Again, with basic organic chemistry, you know that because of the fatty acids, and the fatty acids are comprised of long chains of carbon atoms, you know that this part of the molecule here is non-polar. And because it is non-polar, this is hydrophobic, meaning that it does not interact well with water. Say a water molecule comes in with two charges. Let's say I'm going to draw my water molecule here with a negative charge and a positive charge. Comes here, does not see any charge to interact with. Only in this side, in the head, where you find a positive and negative charge. Therefore, it will not be able to interact at all. That's why, because you find a hydrophilic and hydrophobic side on only one phospholipid, you call these molecules amphipathic. They're able to do both things. They're able to be hydrophilic and hydrophobic, like amphibians who are able to walk on land and live in water. So these molecules are able to do two things, two opposite characteristics. Okay? Another thing I want to mention is that the main component of the cell membrane is phospholipids. So if you look at a cell, again, my beautiful cell here with a nucleus, this area here enclosing the cytoplasm, which is the cell membrane, is comprised of a phospholipid bilayer. And by phospholipid bilayer, you're going to learn that in more detail. I'm going to show you in a different tutorial. But a phospholipid bilayer, you see all of these structures, but in two layers. So this is why. Bilayer. Again, word origin. And this is what you find in this area here in the cell's membrane. And you will also see other structures like proteins and carbohydrates, but the main component is definitely phospholipids. Second thing to mention to talk about phospholipids is that they're important in signal transduction. Signal transduction is when something happens in the environment and say there is stress caused or something is happening, there are signaling molecules in the environment which will interact with the cell's membrane, and then it will produce some sort of signal, and therefore something is going to, like a pathway is going to be ignited, let's say, or stimulated, let's say, inside of the cell. So if you imagine a molecule interacting with the cell, the first layer that it's going to find, the first thing that it's going to have contact with is definitely the phospholipid bilayer, and therefore there is some importance involved in the signal transduction, and we are going to see that later on. But for now, keep in mind that phospholipids have or play an important role in signal transduction. I want to give a quick note on this one. We're still talking about phospholipids, and the molecule, we have two molecules here that need to be compared very briefly, and the molecule you have on the left is a glycerophospholipid, like the one I talked about previously. So this is no need for an S. So this is a glycerophospholipid, meaning that I have a glycerol molecule here bound to a phosphate group that you find here. So this is my phosphate. But there is another type of molecules that you can find in your cell's membranes, and these are called, they have an interesting name, sphingomyelin. These are found in your cell's membranes, so other types of phospholipids found in your cell's membranes. And what happens here, the major difference between these two, you can still find a polar head here because you have a positive side here and a negative charge here. So positive charge and negative charge makes that polar head. But instead of glycerol, you do not find a glycerol here. Instead, you find a, let me change to red. Instead, you find a serine molecule. And this is where your fatty acids are attached to or bound to. And what happens here is that if you look at this structure here, so this structure here is called, trying to draw here, this structure here is called a sphingosine. I'm just giving you nomenclature and explaining a little bit why you find, or the importance of sphingomyelin in your cells. Quickly, do you find them? It's a phospholipid, and the major difference here is that you do not see a glycerol molecule. Instead, you find a serine molecule, and this is where your fatty acids will bind to. Another class of lipids that we see out there in your cells are these called glycolipids, and glyco means carbohydrates or sugars. So you're going to find a carbohydrate or a sugar associated to a lipid. And as you know, sugars are good solutes. They dissolve very well in water, and therefore if you associate a carbohydrate to a lipid molecule, what you're going to do, you're actually going to be able to increase the solubility of the molecule, because as you know, lipids do not dissolve well in water. They're not good solutes in water, but if you associate a carbohydrate, you can definitely increase that solubility. The second point I want to make on glycolipids is that they are secreted to or located in the cell surface. And what I mean by this, if I have my cell here again, you can find these molecules somewhere on this area here. So the outside of the membrane, okay, located on the outside, not the inner part that is in contact with the cytoplasm, but the outside of the membrane that is in contact with the outer environment, okay? This is where you find glycolipids. And it actually is associated to the function of these molecules because they work as markers, or they serve as recognition sites in cell-to-cell interaction. So they work as markers. And what this is that these molecules are able to work or to be some sort of code, I would risk to say, that is used in this type of interaction. And that's why we call it markers. Now let's talk about steroids. Steroids are definitely lipids, okay? And they function as signaling molecules in your cells. And I have a basic structure of a steroid, which is the sterin structure. They have this common structure. And what happens is that a sterin structure is comprised of 3 cyclohexanes, meaning 3 6-carbon rings. So 1, 2, and 3. 6 carbon rings. And 1 cyclopentane, meaning 1 5-carbon ring. And this is my 1, so I'm going to put 1 cyclopentane with 5 carbons, so a 5-carbon ring. And examples of steroids, you can find them in many forms, but they all have this sterin structure, keep that in mind. But the forms or the examples you can find, steroids are cholesterol, steroid hormones, for example, testosterone is a steroid hormone. You have testosterone, is a steroid hormone. And bile acids are also steroids or have steroids, and vitamin D3 is also part of this group. The last class of lipids that I want to talk about in this tutorial is the carotenoids. And carotenoids are only found in plants. Some bacteria have them, and some fungi. but you do not find these in the animal kingdom. We are not, humans for example, are not able to produce carotenoids, but we can actually obtain them through our diet and use them for some metabolism functions in our body, but we're not going to discuss that now. Just keep that in mind. And one thing that is important to refer to when we talk about carotenoids is that they play major roles in photosynthesis, and also they're responsible for some pigments. And the pigment is actually a result of the structure of the molecule. And I have an example here. This is beta carotene. And I want to use this molecule to explain a quick thing, an interesting fact about carotenoids, which is conjugation. Okay, tongue twister right there. Conjugation on the carbon-to-carbon double bonds. These double bonds that you see here in red. This is a very important structure because these conjugated bonds allow electrons to move freely throughout this molecule. So that's an important characteristic of carotenoids. Another thing I would like to say quickly, and I'm gonna try to make this molecule fade out, and are the examples of carotenoids that you find, especially in humans, that are useful for humans. It's beta carotene and retinoic acid. So keep that in mind.